Welcome to the online dairy education series, or welcome back. I know a lot of people have been attending all the webinars, which has been great. We've definitely had fun, had a great lineup. I've learned a ton myself. So thanks everybody that's been participating. And just want to thank USDA NIFA and USDA SARE and Risk Management for their ongoing support of our dairy education in the Northeast, which has been really great. Today, we're going to be hearing about local and regional research projects that are focused on dairy farming, and lots of different topics, a pretty good span of topics here. And today we're being joined by Dr. John Barlow, who's an associate professor in animal science at UVM, and he conducts microbiological research related to dairy cattle health and disease. He's going to be talking about some bedded pack research today. And then after John, we're going to hear from Dr. Andre Brito, who's an associate professor. I thought you're associate professor, Andre, or are you full professor now? I can't keep up. In UNH's Department of Agriculture, Nutrition, and Food Systems, where he studies maximizing dietary nutrient utilization from forages in dairy cattle. So we've been hearing a lot about that. And then Miriam Snider, a graduate student, a PhD candidate here at UVM in the Department of Animal Science, will be talking about her research in forage digestion kinetics to optimize production in high forage dairy cattle rations. So again, you can see lots of conversation about feeding lots of forage. And then finally, we'll hear from Dr. Kathy Soder, who's an animal scientist at the USDA-ARS in Pennsylvania. And she's been conducting research on pasture-based systems for many years and grazing behavior to maximize forage and animal productivity. But I think today she's going to be talking about interseeding cover crops. So we'll see. It'll be exciting to hear from everybody and get a little update on what's going on. So our poll is up, and thanks everybody for answering it. It looks like most people use local research, which is great. Part of why we want to have these types of webinars is so that we can get that information out to you. And we continue to try to do that in various ways. All right, so John Barlow, with that, I will turn it over to you, and I'm marking down the time so we can try to stay on time here. And I'll let you share your slides and give you the floor. So if you have questions for the speaker, please type them in the chat box, and we'll try to take a question or two between speakers, and hopefully we'll have a few minutes at the end to take any final questions. All right, we can see your slides. John, take it away.
Thanks very much, Heather. Hi, everybody. I'm really happy to be here and give you an update on our USDA Organic Research and Extension Initiative project, which was looking at bedding strategies for promotion of udder health and milk quality on organic dairy farms. And we're particularly interested in doing microbiome work, but I won't tell you about any of that today because that's still pending. But I will give you a little bit of background on the basic epidemiology of mastitis that we're seeing on the organic dairy farms that we've enrolled in this project. So the overall objective of the project is to look at the relationships between how we're managing cows in the winter on winter bedding systems, and how the microbes that we find within the system, humans, cows, the bedding, the manure, how they interact with the teat skin and the teat canal, and potentially the intermammary tissue. We're and causing intermammary infections. And we're interested in both the pathogens and the beneficial commensals, the potential beneficial bacteria that may be in the system and may actually contribute to resistance of cows to mastitis. This concept that the microbial flora of their teat ends or their microbiome of the mammary gland may actually contribute to a cow's ability to resist infections or have a resilient udder to resist infections. So the first thing that we did in this study that has been published just recently, accepted for publication in the Journal of Dairy Science, was a survey of the industry to understand what type of winter housing and bedding systems were done on Vermont organic dairy farms. And big thanks to everybody, and it's a lot of you, who returned the survey. We had an 82% response rate. 145 farms returned our survey from the approximately 175, 180 organic dairy farms in the state, which is spectacular. And overall, it shows you that the dominant housing system, approximately 40% or more, is tie stalls, cattle housed in tie stalls with sawdust or wood type shavings bedding systems. But there's a range, and we are particularly interested in understanding how the bedded pack systems behave and comparing that to the dominant systems in the state. So our study designs subsequent to this survey are looking at the teat end and milk microbiome and mastitis risk on freestall herds that are bedded with wood products, freestall herds bedded with sand products, and freestall and tie stall herds bedded with wood products, and comparing that to the bedded pack herds that we have in the state. So we have a couple different observational studies that we're doing that with. And one of them is what we call a longitudinal study, where we sampling cows over time. And for that study, we enrolled 5 bedded pack herds and 5 tie stall herds. And from each of those herds, we collected randomly selected cows, we stratified cows that were low days in milk, 35 cows per herd, so that we could follow them over time from December to March, 3 months we ended up with 3 months sampling per cow or per herd. We had wanted to do more, but you know what happened in March 2020 with COVID, and that restricted our ability to complete this study. But overall, we collected a little over 4,200 quarter milk samples and teat swabs from the 1,500 quarters of 384 cows on these 10 herds. Completed aerobic culture and quarter milk quarter SCC, somatic cell count of the samples. We have DHIA records from most of the farms. Completed teat swabs processing for genetic analysis, and quarter milk processing for genetic analysis is ongoing right now. Preliminary results from that study, looking at the mastitis epidemiology, we have about 60% of the isolates that are causing intermammary infections based on increased somatic cell count or being able to culture the organism from multiple samples taken at the same visit, from 2 samples at the same visit. The majority of the most common pathogen are these non-Aureus Staphylococci. So all the other Staphylococci that group in this one sort of group, which is probably shouldn't be treated as a single group, all those other Staphylococci that are not Staph aureus. So you can see the Staph aureus results here. We have about 12% of the intermammary infections due to Staph aureus, about 15% of intermammary infections due to Streptococcus or strep-like species, and the dominant organisms there are Strep uberis and Strep disgalactiae. So they are environmental source primarily, and that will be an interesting group to look at as we tunnel down into understanding these data. And then the majority are these non-Aureus Staphylococci, which we really don't know. The industry doesn't really know a lot about the epidemiology around this. And we think that they are there's about 20 or 25 different species that contribute to this group, and they are probably a mix of organisms that could have beneficial effects on the teat skin, as well as potential pathogens. The potential pathogen, so I'll skip ahead and talk to you about the potential pathogen in a moment. For now, we can look at the somatic cell count results of those organisms. So on the x-axis here are our organism groups, the Non-Aureus Staphylococci, Corynebacteria species, no intermammary infection or no pathogen isolated, the majority of our quarters, Staph aureus, or these Streptococcus species, Strep uberis, Streptis galactiae. And if on the y-axis, if we look at the linear score, so a conversion of the somatic cell count score, where a linear score of 4 is equivalent to 200,000 cells per ml at the quarter level, we can see what proportion of quarters actually exceed that threshold for having mastitis or an inflammation in that quarter. And so there's a lot of diversity. Some of these CNS species aren't causing any pathology or any inflammation when we collect them from the quarter, and some are significant. Maybe some of them as high, causing as high a somatic cell count as Staph aureus does. But you can see these other, what are classically described as major pathogens for good reason, like Staph aureus and the strep species. Those isolates, the majority of them cause an increased somatic cell count above our threshold of 200,000 cells per ml. So we would really like to target these species for sure in mastitis control. And at this point, we don't know what to do with these species in mastitis control. We'd love to target these infections that are high somatic cell counts, but the rest of them may be inconsequential and may actually be beneficial to the cow. As we think about that, we think about how we look at bacteria. And so there's a hierarchy. And so most of you are probably familiar thinking of bacteria at the species level, distinguishing Streptococcus species like Streptis galactiae, Strep uberis, or Staph aureus, or E. coli. But our work now, we're moving to the strain level and being able to, for example, with Staph aureus, distinguish Mrsa from non-Mrsa, or penicillin-resistant Staph aureus from penicillin-susceptible Staph aureus. Those are, you've probably heard of Mrsa, methicillin-resistant Staph aureus, really important pathogen. And we use genetic techniques to do that, to distinguish these strains. We've moved now to do that with the most common Staphylococcus species that we see in our sampling on dairy farms. And that's one maybe you haven't heard of yet. It's called Staphylococcus Chromogenes. And of the isolates that we've looked at right now, about half of them where we've speciated the Non-Aureus Staphylococci, 75% of them are this Staph Chromogenes. So the majority of the CNS, the Non-Aureus Staphylococci that we're seeing on farms, are this species Staph Chromogenes. It seems to be a mammary adapted, cow adapted organism with some pathogenic potential. We've developed a genetic sequencing system that will allow us to distinguish between the strains of Staph Chromogenes, just like we can with Staph aureus. And we can use that then to think about the source of those, the source of that organisms, whether we're finding them on cows associated with intermammary infections, or just on the teat skin, or in the milking system, which could come from either source, cow skin or teat skin, or other places, the environment, or whether we find them in the environment. And in this study, which was a previous study that we did, we also included an occasional pig when we had a pig on the farm, and found Chromogenes from those pigs. So the majority of the Chromogenes that we're finding are from intermammary infections, the bright green. We rarely find Chromogenes in the environment. So that supports to the idea that perhaps Chromogenes acts a lot like Staph aureus, where it's spread from cow to cow. Further study as we go along, we're going to think about this. Instead of thinking about the source, we'll then think about the characteristics of these staph Chromogenes infections and whether they're, for example, more virulent, if you will, as measured by somatic cell count, whether those staph Chromogenes organisms cause a higher somatic cell count or not, if we can find strains that seem to be more pathogenic, if you will, by causing an elevated somatic cell count in the cow, or alternatively, if they have a longer duration of infection, that they tend to be strains that tend to be associated with chronic infections in the cow. So we've done this in the past for Staph aureus infections and contribute to understanding of the epidemiology or transmission and importance of Staph aureus within the cow. And now we're going to move and do this for what seems to be emerging as the next, if you will, barrier to milk quality, this staph Chromogenes intermammary infections. The other thing that we can gain from this longitudinal study where we're sampling the same cows over time, so longitudinally repeated samples at the same cow, is we can characterize their intermammary infections based on the duration of infection or their transition. So we can look at chronic infections, cows that have the same organism from the same quarter for all 3 months. And these are definitely the cows that we would want to target for control, to do something with, to eliminate that infection. Because in the case of Staph aureus, they might be the source of a infection for another cow. Or cows that had transient infections where they became infected and perhaps stayed infected, or became infected and recovered. And these then we'd sort of understand that these cows are more resistant and to their the pressure from the pathogens within the herd. And we'd like to understand the microbiome, their teat skin microbiome, what makes these cows different from these cows that are chronically infected, and target those differences. And it may be associated with the microbial flora that they of those teat skin. And then compare those also to the never infected cows, because these are the cows that we want, right? That seem to be resilient to infections. So that's the work we're headed for. One tool that we're using from these teat skin swabs that we're taking, where we take swabs of the teat ends and look at the microbial flora on the teat ends. One tool that we're using, and we did a pilot study in the UVM teaching herd, is a study that's called simultaneously antagonism study. So we do culture work where we take the teat swab samples and plate them on one side of the plate, and then plate the next day and culture that. And then the next day we plate Staph aureus on the other side of the agar media, and we look for organisms from our original teat swab sample that inhibit the growth of Staph aureus on that plate. So you can see here's an example of an organism on the other side of the plate that's secreting something that prevents the growth of Staph aureus, sort of a natural antibiotic. These natural antibiotics are often called bacteriocins, and here's an example of an organism streaked in a straight line on the other side of the plate. And here you can just see this little bit of zone of Staph aureus on the outside. This organism is a really strong inhibitor of Staph aureus. So we're looking from the teat swab samples for these types of organisms and trying to understand what cows have them, what bedding systems may have more of these natural inhibitors of pathogens. So that's what I have for today. I want to thank all the people that have been involved in this study, our research team, our scientific advisors. We have a great scientific advisory team for this project, and our farm advisors. So we have a really great farm advisors. And of course, I can't fail to mention the collaborating farmers. They've been integral to this, both for the study that I described, that 10 herd study, as well as we have a larger 40 herd study that some number of farmers are engaged in. Again, our funding source is from the USDA Organic Research and Extension Initiative, and we have one more year of that funding to go, and we're really excited about the next steps looking at the actual microbiome and the endogenous inhibitors in the system. If you want to check in with us, Dr. Neer's Twitter account is @SoilSuccess. My Twitter is @UVM Barlow Lab, and Barlow Lab also has an Instagram account that we just started up. So thanks very much, and I think we've got some time for questions.
Yes, absolutely. Are there any questions? Yes, there are already questions. Okay. Have you observed significant cases of Mrsa on organic farms here in New Hampshire?
So we've only looked in Vermont, but we have looked, and thanks for that question, because we've looked both in organic herds. We had about a couple years ago, we did a study where we looked at bulk tank milk samples in organic herds. And then more recently, we did a study on artisan cheese herds, herds that have cheese plants on their farm. There were about 20 herds in that study as well. And the good news is no methicillin resistance in those surveys. So can't say that Mrsa doesn't exist in our farm systems, but we do know that from those surveys that at least the prevalence in those farms, there's no evidence that it's there. The cool thing about the artisan cheese farm study is we also swabbed people on the farms, and we found no Mrsa in the humans on the farms, no methicillin resistant Staph aureus. We did find one methicillin resistant other species. So that's a concern about whether some of these other species like Staph aureus may act as a reservoir for the antibiotic resistance genes to transfer, if you will, to their cousins, like Staph aureus or the other staph species. Thanks for that question. So far, no Mrsa. Really great.
Great. Wondering about comparison between NNN cows and NYN cows for capacity to recover from infection as a desired trait, and any differences in testing results.
Yeah, we haven't gotten there then. So I just gave you a little sniff at that sort of thing, but that's exactly where we're going. We're going to do it from the perspective of the microbiome. Don't have any data relative to the perspective of the cow itself to say the genomics of that cow, any information on that, other than what we might glean from DHIA data. So for the farms that are on the study from DHIA. So we haven't explored that fully yet. We've just identified the number of cows that fit those categories. And now the next step is we're going to look at their skin microbiota, their skin microbial flora, and see if there are any major differences in the communities that those cows harbor. And of course, we'll control for the type of farm system they were from, whether they were from a tie stall herd or a bedded pack herd. Thanks for that question.
All right.
That's
Okay. Yeah. Well, thanks, John. I am going to let you off the hook here, and we'll see if we can get Andre. If you could share your screen. Thanks for staying on time, John. That was perfect.
Yeah, thanks very much. That was awesome.
And
I really appreciated the opportunity, Heather. Thanks a lot.
Yeah, don't worry. We'll get you back for more updates. All right, Andre, you want to, let's see, looks like there you are. I think you, yep. Perfect.
Good.
All right.
Yes, changing a little bit of the subject here. But that was a great presentation by John. So we'll be talking about feeding legumes to organic dairy cows. And thanks, Heather, for the opportunity. Always great to contribute here. So I will start with research that we did not too long ago, looking at actually Heather groups are also part of this research. Basically, we collect samples, pasture samples across 14 organic dairies in the Northeast from Maine up to Pennsylvania and also New York. And what we basically noticed there was there is a large variation in net energy of lactation, NEL, right? So you can see that there is year variation, month variation, and also farm variation, minimum from 0.35 up to 0.80 megacal per pound. This is basically on over 380 samples of pasture. So it's not, I don't think it's any news here that net energy of lactation energy is one of the limitations on high forage diets, right? So there is ways that people could capitalize on high forage diets and trying to improve the energy on those forages. And this has been done in different ways. One way that could be done is it's taking advantage of the photosynthetic capacity of plants, right, to accumulate sugars. And this is one way to that can be done via the PM cutting, for instance, or hay in a day kind of management strategy. And this data here is basically shows like exactly that. So you see in the x-axis is the time after sunrise, right? And your y-axis here is the carbohydrates. And the carbohydrates are breaking down here into starch, soluble carbohydrates or sugars, and the total there is NSC or non-structural carbohydrates. And you can see that as the day pass, right? The accumulation of sugars occur. And then by 13 hours or so after sunrise, that's the higher concentration of sugar. So that's where it could be cut, right? This is done in alfalfa in Canada. So this here in Quebec, that the spring growth, and then you can see the regrowth of that same field, and you can see the same kind of behavior. So about 12, 13 hours after the sunrise, you have the accumulation of sugars, and this can then increase energy in those forages. We did, actually this study was done in Canada a while ago, but in this study we looked at cows that were fed either PM cut, alfalfa baleage, or AM cut, right? And you can see here that, and that's sort of the only forage source. These cows did not get any grain in the diets, right? So you can see that both dry matter intake went up and milk production went up. Not much. So overall was about 5% increase. This was statistically significant, but you're talking about here 2 or 3 pounds difference, right, in milk production and intake, but seems to be a response there. Not big response, but there is a response in diets that were 100% forage based, right? So when I look at milk fat, that's the same study. Again, the PM here in blue and the in yellow, this color is, but anyways, you can see that both, again, milk fat went up by about 10% and milk protein also went up by 7%. And that's probably related to the increase in intake, right? And another thing that's interesting on this data set is microbial protein. I think was discussed here last Tuesday, the importance of microbial protein, right? Trying to maximize microbial protein in those diets as much as we can based on the good amino acid profile of microbial protein, relative to the cow needs, specifically to produce milk protein. And you can see also there was about 7% increase in microbial protein in those diets that were, those cows that were fed the PM cut baleage, right, as the only feed source in those diets. So yes, there is a response there. Also, there is some variation in how we can get these sugars going up, from year to year, but it is an option out there, right? So we also know that this is work done by Seed a while ago also, that you need to keep a certain level of legumes in those fields. These were done with pasture samples, and it seems to be a threshold there of about 30% or so. So looking this data from Seed, right, where that 30% of legumes in the pasture it's basically when you start to see a drop in fiber, right, in components of the forage that less digestible, and while you see an increase in protein and also other compounds, including net energy and TDN, right? It's not showing here, but that's kind of in the same data set, they show that, right? Our data indicates also a lot of variation. This is that same study that I shared earlier, where we collect samples over 3 years across 14 organic dairies, and we did some visual observation of those pastures and came up with ways to quantify, right, the proportion of legumes in those fields. And you see, again, a big variation, right? So it's not a surprise here as well. So there is variation by year and also a trend here for a month by year interaction. But you can see that, you know, potentially, not potential, there is data here showing that was below that 30% threshold in some years, or a little bit up. But what I'm trying to say here, that persistency of legumes on pastures and hay fields has been difficult in the region, right? Another thing that I want to show why kind of it's important to have legumes in the diets. So this is a data set, is a meta-analysis that is very powerful because it combines lots of studies. And they compare basically grasses against different legumes, white clover, red clover, alfalfa, and buzzfoot trefoil, right, in this meta-analysis. And what they saw here was overall legumes perform better in terms of intake, milk production, energy-corrected milk, except digestibility here that seems to be lower on legumes than grasses, except white clover. But overall, legumes were better than grasses. These are head-to-head comparison in high forage diets, and specifically buzzfoot trefoil really improved milk production and intake, right? Followed by white clover. And red clover and alfalfa were intermediates there, but all of legumes, you know, led to better milk production and intake on those cows. So again, important to have legumes, and that's kind of motivate our group here in collaboration with UVM again, work with seed and specifically we conduct this study looking at several aspects. I think seed shares some of this data Tuesday, and I'm sharing here today more of the cow data. These studies were done at the UNH Organic Dairy Farm, right? Some of the methods that we use in this trial, I'm going to show 2 trials here. We use 20 organic certified jerseys, 10 cows per treatment. Either those cows get alfalfa grass or red clover grass mix in the diets. These were done in a randomized design with a 7-week length of the study, with samples being collected on week 4 and 7. That's how I'm going to show the data. We also collect methane, and I will share a little bit more about methane as we move along here. So just to have an idea, these fields are set up as to be 80% legumes and 20% grasses, right? So, but in fact, things were a little bit different than what we planned. It's not a surprise here as well. You can see we use alfalfa in cut 2 or cut 3, alfalfa in red clover, right? So you can see the cut 2, we have about 60% alfalfa, 70% red clover. We're supposed to have more grasses here. We expect to have about 25% of grasses, but you can see there's more weeds also in one of the cuts. In the cut 3, you basically lost all the grasses and the legumes really took over, right? And some of the weeds, at least in the alfalfa field, is there almost no weeds on the red clover. So what I'm trying to say here, overall, you'd have a field that was predominantly alfalfa or red clover, right? So here's the nutrition composition of those balers. I'm gonna highlight a couple of things here. One is soluble crude protein. This was discussed by seed as well. You know, red clover usually has less soluble protein compared to alfalfas, and this can and have an effect on nitrogen use efficiency, and I will show that a little bit from now. And another thing was starch and sugars. It seems that those red clover would have more sugars, as you can see here, compared to the alfalfas, and alfalfas will have more starch. So there's that switch there. But overall, red clover seems to have more sugars there than alfalfa mix. And here are the diets. We fed a 65-35% forage to concentrate ratio. Either the diets with alfalfa only obviously has alfalfa, the red clover only red clovers, and we have this 35% mash, grain mash there based on soybean and ground corn. And you can see here the protein of the diets and the NDF of the diets, relatively similar, right? So when you look at data, right, dry matter intake, we didn't see any difference on those. You see alfalfa in blue here and red clover in orange. So intake was relatively similar here, not statistically different. The same for milk production, no difference. Doesn't matter if you fed alfalfa grass mixture or red clover grass mixtures in this case. However, when you looked at milk fat, was a little bit better for the alfalfas, right? So roughly those cows that get alfalfa produce more milk fat, about 9% more, right? This is something that's interesting to look at, but no other difference in terms of the milk components other than milk fat yield. When you look at MUN, that's what I said before about red clover. Red clovers has less soluble protein, right? And also has an enzyme called polyphenol oxidase that prevent breakdown of proteins, not only during the silo process, but also in the rumen of the animal. It binds to proteins and prevents degradation of protein in the rumen. So that leads to less urea circulating in the blood, right? And less urea that goes to the mammary gland as MUN. So you can see here that MUN dropped for about 16% in the cows that were fed red clover. There's a typo here, red clover here. But anyways, so that's consistent with cows that are fed red clover. A better nitrogen use efficiency. When you look at fatty acids in the milk, right? We saw some response here. Cows that were fed red clover, they have more of this trans-10 fatty acid. This trans-10 fatty acids is related to milk fat depression and reduction of milk fat synthesis. So maybe that's partially explain why those cows on red clover has milk fat production. We didn't see any difference on the CLA between diets. They're similar. However, when you looked at the omega-3, the alpha-linolenic acids, the major omega-3 present in milk, those cows that were fed red clover had high concentrations, right? High content of those fatty acids in the milk compared to the alfalfas. Also, the sum of omega-3, that's the total omega-3 fatty acid, was higher on the red clovers. There is an interaction here, but basically the difference was more on week 3 than week 4. But again, overall, red clover produced more of those omega-3 fatty acids, and they also had a better omega-6 to omega-3 ratio. It's lower, right, on red clover. If you think about the ideal would be 1-to-1, right? And organic milk usually has a lower, right, omega-6 to omega-3 ratio. This one's relatively low there. So again, so that's changed in milk fatty acids more favorable towards the red clover diet if you think about human health. In terms of methane, one thing that's important to talk about methane here, methane, in addition to be a greenhouse gas that a lot of people are concerned, right? And in general, we all concerned about that. Methane also represent energy losses. I'm not expecting here, we're not expecting necessarily change in methane in terms of we feed those diets, thinking in changing methane necessarily, but we're looking more into the energy balance of the animal. So more methane means more energy losses. We want to understand more what happens in those cows fed these high legume diets. And even though we see here a drop in methane on week 4 on those red clover diets, we did not see that on week 7, right? So there's this interaction here between the weak and the forage source. So basically it was inconsistent response in methane in terms of energy loss of those animals. When you express methane in grams per kilogram of intake, of DMI, of dry matter intake, there is no difference. Also no difference in when expressed grams of methane per kilo of energy corrected milk. So overall, we could say that these diets are relatively equivalent in terms of energy losses, right? So so moving on now to the data from the second study, this we basically use also 20 cows, a slightly different experimental design. It's a kind of a crossover design. In those cows, again, were fed two diets, either a red clover grass mix, and in this time, we also fed a diet with alfalfa white clover, right? So that's a slightly different mix of legumes here. And we also measure methane emissions here. And we have 14 days where the animals are adapting to the diet, and then followed by 10 days of sample collection. Then we switch those diets across those animals. Here's the same field and how this field looks a year later, right? Exactly the same field. So see, can major change, some change here. So now the red clover grass second cut still relatively consistent there in terms of red clover. Didn't lose much red clover here. However, the alfalfa has dropped a lot. We have a lot of winter kill of alfalfa, and some of that alfalfa was replaced by white clover, as you can see here, right? So those are the forage source that we used in the second trial. So when you compare those baleage in terms of their nutritional composition, again, you see the red clover has less soluble protein because of that enzyme polyphenol oxidase that's only present in red clover. And then same response, red clover has more sugars, as you can see here, and alfalfa more starch. That was consistent across these two studies that we did. Here's the diet. So in this case, we have this red clover grass mix, 60% of those baleage in the diet, second or third cut. And we replace some of that, right, with this white clover grass mix. So the second diet have red clover and then a mix of alfalfa and white clover, almost like a 3-legume grass mix, right? And diet is about 18% crude protein, about 30% NDFs. When you look here, mostly did not see any difference in production response, other than dry matter intake was slightly higher, statistically significant, and higher for the red clover grass mix. Few changes here, a little bit more milk protein in the 3-legume diet, but this is mostly driven by the lower milk yield, so kind of concentrate that milk protein there. And then the PUN tend to be lower, that's plasma urea nitrogen, tended to be lower with the red clover. Again, that indicates that in general, red clover, because of that enzyme polyphenol oxidase, leads to better nitrogen use efficiency. MUN drop also, but not statistically, right, as you can see here. So one thing is interesting, digestibility. We measure digestibility in this study, and those cows that receive the red clover grass mix, they have a better digestibility, right, of dry matter, organic matter, and fiber, NDFs and ADFs. However, this more digestibility did not translate in more milk production, as you said. It translated more intake, a little bit slightly higher intake, but not in more milk production. When you look at energy losses using methane here, as you can see, we also, I forgot to mention that for measure methane, we use this equipment here, the green feed, that basically is a head chamber. The cows put the head in, and while she's breathing and belching, you know, there are sensors that will quantify methane and all the gases, and also CO2, hydrogen, and just focus on methane here. But you can see here at the bottom with this figure. So while the cows there, to entice the cows, there is a baked grain there that it's released automatically in the cows while she's consuming that grain, the pellet about, you know, in the day will be less than a pound. So basically we get the cow several times there, right? So, so you see there's no difference in methane production. Statistically, in methane yield, in grams of methane per kilo of dry matter intake, so there's a bit higher in that 3-legume grass mix. That kind of shows they were losing a little bit more energy if you express on a dry matter intake base, but when you express on an energy corrected milk base, there is again numerically higher, but not statistically significant, right? So what are the take-home messages here? We see that overall increasing the proportion of legumes in dairy diets can improve NEL and also dry matter intake and milk production if comparing to grasses. Based on UNH feeding trial 1, we saw that alfalfa did increase milk fat production, and red clover reduce MUN and improve omega-3 fatty acids. These are linked to that enzyme that I mentioned, the polyphenol oxidase enzyme. On trial 2, red clover grass did increase the dry matter intake and digestibility compared to those 3-way legumes there, the 3-legume grass mixtures. However, did not result in more milk and milk components. So with that, I would like to acknowledge here some of the funding source, my lab, my students that work on those studies, UVM for the opportunity with Heather groups there, and open up for a couple of questions if you have time.
Yeah, thanks, Andre. That was great, really interesting. It's always wonderful to hear about all the work going on. If there's any questions for Andre, we can take a question, and if not, we can save them up. I don't see anything yet. I've been hearing this a lot about red clover. It seems like there's so much good about it beyond what we knew. I hear it from a lot of nutritionists too, that red clover seems better for the cow. Is that sort of what you're coming up with? It seems like there might be a couple of downsides, but is that a legume that we should be getting more of out in the fields?
Yeah, I think, I mean, you know, red clover, there is data there that shows that what I saw show here today, sometimes they have better digestibility when you compare to alfalfa, and sometimes that's not translated into more milk production. But when you look at nitrogen efficiency, MUNs and PUNs, that happens. You know, they lead to a better nitrogen use efficiency in general. In terms of production, I would say there's more data with alfalfa that leads to more production, you know, milk production and things like that. That's why I try to combine in that second study, trying to combine the hypothesis was if you combine some alfalfa and white clover that are showing to be, you know, specifically white clover that's showing to be better overall, it will mitigate some of that, and kind of see that happening there. But I mean, I'm interested in red clover a lot based on the nitrogen aspect and less nitrogen excretion to the environment. So I think for that, for sure, it's
yeah, that's great.
Yeah.
Yeah, there's so I don't know, Miriam, if you want to put your slides up, I'll ask Andre another question, but somebody was asking about are there any significant higher levels of NFC in red clover compared to alfalfa and white clover?
I think, you know, based on this and all data, and also based on data that was done in Canada, among several legumes that they test, red clover is the one that comes to the top in terms of NFC or non-structural carbohydrates, basically starch plus sugars. And we kind of show here if how of that is really come up to be significant at the animal level. That's another story, but at least on in the comparison base, yes, ready clover has been on top of those, but then there is difference in terms of, as you can see here, digestibility that we cannot not completely explain why they lead to more digestibility, but not being translated into milk production. And white clover then is definitely one of the best ones in terms of digestibility in the animal. So there is some of those trade-offs, right? But red clover seems to be on the top in terms of NSC and NFC, both fractions.
Great. So I will say, Andre, there is a question in the chat about phytoestrogens, and I'm not sure. I'm going to let Miriam get started, but we can either take it at the end, or if you want to answer it in the chat as well.
I can answer in the chat there,
okay.
so we don't take more time.
Okay, we can always repeat it afterwards
yeah.
too. All right. Well, thanks, Andre. And Miriam,
Sure.
you're ready?
Ready to ever be.
All right. I'm going to turn it over to you.
All right.
Thank you.
Well, is it sharing?
It's sharing.
Okay, there it is.
It has,
all right.
Yep, there we go.
Well, all right. Thanks, Andre, for that talk. It was really great. And actually, mine kind of ties into some of the things that Andre was just talking about. And I'm going to talk to you all today about some of the research we've been conducting, where we've been looking at various forage mixtures on rumen energetics and performance. And we used a method, an in vitro method called continuous culture. So a brief overview, I'm going to give you all some background into our previous research that shows how what drove our current research topics, and I'm going to give you all some materials and methods, results, some ideas moving forward, and then we'll take questions at the end. So we know now that many dairy producers in the northeastern United States, they use these high forage diets on farm, or they're transitioning away from these primarily grain-fed diets, and quite a few of our pasture-based producers around here are now certified organic and grass-fed. And this means that their animals have to be on pasture for at least 150 days, and they receive 60% of their dry matter intake from pasture grasses. And there are a few reasons for this switch, although they are kind of complex. So lower production costs could be one reason, because we know that forages can be cheaper sometimes depending on your circumstances, and they can be an abundant feed source. We also know that there's some improved animal health reasons for switching to a grass-fed dairy system. And there is this perception that there's a better quality of life for both animals and producers. But up until recently, a lot of these perceptions are based on management decisions, but up until recently, there wasn't a lot of information known about why these organic grass-fed producers are making the decisions they do, or what influences their management decisions. So to figure out how to better serve the organic grass-fed community, last year we conducted a study in which we evaluated research needs and management practices of our grass-fed dairy farmers in the US. And this was just accepted for publication with some minor revisions. So we found that the majority of our surveyed producers, when we asked them about their satisfaction of their pasture qualities, such as yield, stored forage quality, soil fertility and health, and their stored forage yield, on a scale of very unsatisfied to very satisfied, we found that when we summed up our somewhat satisfied, satisfied, and very satisfied categories, that the majority of our producers were happy with their pasture qualities, and those that were satisfied also indicated that they had greater milk production. From the same study, we asked these same producers to tell us about what they thought they knew about their forage parameters, such as growing higher energy forages, or how to improve their forage quality and some of their energy requirements for their animals. And this was also on a similar scale. They ranked their knowledge from very low to very high. And then we added up our average high and very high to get our response variables. And what we saw is that about 83% of our producers said that they knew at least an average amount about growing higher energy forages, while 92% said that they had at least an average knowledge of energy requirements for their animals. So while these are great numbers, there appears to be this kind of slight disconnect. And what's concerning here is this 15.8% that says they have a low knowledge of growing higher energy forages, because that changes when we get to our energy requirements for cows. So this means that some of our producers, they don't know how to grow high energy forages, but they think they know quite a bit about what their animal needs. So what we could say is that there may be these results, they're not getting the results they want based on pasture quality and yield satisfaction, and maybe they don't know as much as they perceive that they do. So that does beg the question, how are our producers meeting animal energy demands adequately? And what can we do to help them grow forages to meet their animal energetic demands? So to answer that question, we started by developing some research questions about what's actually in a typical northeastern pasture and what we can how we can best utilize what's available. So if you attended Sid's talk on Tuesday, he touched a bit on pasture diversity, but we know that pastures are typically composed of a diverse number of forages, and that includes your legumes like clover, alfalfa, and burstfoot trefoil. And they can contain grasses, both cool season and warm season. I'll touch on warm season grasses here in a little bit. Sometimes there will be forb species like your ever-present chicory, and then other times you'll have some weeds like, bed straw. And because of this diverse number of forages, there's this potential for there to be a diverse amount of plant secondary metabolites, which can affect animal performance, but that's obviously not the whole story, because diverse pastures are going to have a diverse energetic content. So they'll have things like variable protein and water-soluble carbohydrates. And so while diverse pastures and pasture-based dairy is becoming increasingly popular, there's not a ton of information out there about the use of diverse forage mixtures on dairy nutrition and rumen performance. And those studies that are available, they've been primarily ecological based or conducted in really small plots. And there are some animal trials, but they've produced mixed results. So we attempted to bridge that gap with an in vitro study, and our objective was to evaluate rumen energetics and performance metrics using continuous culture systems that are receiving different forage mixtures. And we hypothesized that a more complex forage mixture will provide energy that's more effectively utilized. So moving into our forages of choice, we chose a cool season perennial orchard grass. And we chose a couple of legumes like red clover again, and alfalfa because they're commonly seen on pastures, although alfalfa is not exactly a pasture species, but you can see it out there sometimes. And red clover we chose similar to what Andre was talking about because of its secondary metabolite and enzyme profile. We also chose to include sorghum, Sudan grass, and pearl millet because they are warm season annuals and they're becoming increasingly popular up here because they can extend that summer growing season when cool season forages begin to taper off in quality and quantity during that summer slump. And all of our forages were grown in late spring to early fall of 2019, and we harvested them at the first and second cuttings and some at third if applicable, and we grew them all as monocultures before we hand mixed them and we fed them to our fermenters. So moving to the animal side of things, we collected rumen fluid from 3 lactating Holsteins from their cannulas here after morning feeding, and we collected from different parts of the rumen because each part of the rumen has a different microbial profile. And after we pooled this fluid into a pre-warmed container, we took pH measurements and we took it back to the lab and we inoculated these fermenters with about 3 liters. And if you're not familiar with what a fermenter is, you can kind of think of it as an artificial rumen. So ours is a dual flow, meaning we have outflow from a spout here and we have outflow from a filter that comes out of here. You can't see it because this one's obviously cleaned up for now. We continuously heat these to mimic rumen conditions at 39 degrees Celsius, and then we agitate them at 70 RPM with an up cycle of about 200. So because these guys do stratify themselves out like a rumen, so you have to mix those contents. They do receive a constant supply of carbon dioxide to maintain the anaerobic environment because it keeps the microbes happy. And then we continuously record pH, temperature, and agitation. So this is to give you an idea of the treatment layout. We laid this out as a 4x4 Latin square design. And what we did is we randomly assigned a treatment to a fermenter. So here, for our first period, we would assign one of our mixtures to this fermenter, and then we'd rotate to the second fermenter, then the third, then the fourth as the periods progressed. So each fermenter received a different forage mixture to account for that variability. And on a dry matter basis, we had 4 treatments that were about 131 grams of dry matter. Our control treatment was this orchard grass alfalfa mix. It was 50-50. Our second treatment was that base control diet plus red clover. And then our third was included millet, and our fourth included Sudan grass. And so to mimic our intake patterns, these guys were fed 4 times a day with 2 feedings at 33% dry matter, so at 6 a.m. and 6 p.m., and then 17% dry matter at 7:20 a.m. and 7:20 p.m. This isn't an all-encompassing list of what we were interested in, but we collected numbers for everything from rumen pH, because we know that our pH is going to be increased with an increased dietary forage, basically because of its relationship with our VFAs. And our VFAs of interest for acetate, propionate, and butyrate. In a forage diet, it's typically a 70-20-10 ratio. We are also interested from an energetic perspective in ammonia and microbial protein synthesis. So we know that microbial protein synthesis requires this balanced supply of non-protein nitrogen, amino acids, ammonia, and energy. But a lot of traditional forages don't provide these components because peptides and ammonia from soluble leaf proteins are rapidly released by proteases. So to measure this, we supplemented our fermenters with an N15 to follow that, an N15 compound to follow microbial synthesis and nitrogen flow. We also were interested in our water-soluble carbs because an increased water-soluble carbohydrate concentration may increase a readily available supply of energy, and this can improve our energetic balance, as well as our microbial protein synthesis, reducing nitrogen losses. And finally, we were interested in methane because there's this relationship, according to literature, that an increased water-soluble carbohydrate content leads to a decreased methane content. And we also collected for proteomic data, so that could tell us what microbes are there and what they're doing and how they change according to diet. And we also collected for polyphenol oxidase, because a lot of our, specifically because we wanted to see what was happening with red clover and if that affected our microbial protein in any way. So these experiments are 10 days each. We start on day 0 and they run through day 10. And on day 0, so day 7, day 0 through 7 is an adaptation period, and days 8, 9, and 10 are a sampling period. And on day 0, we inoculate these fermenters with rumen fluid, and we also give them what's called clarified rumen fluid, which is just a buffer mixed with rumen fluid to help them to adapt to their new environment. On day 5, we added that N15 supplement, and we continued adding that to our buffer after replacing urea for the remainder of the period. On day 7 and then day 7 through day 10, we placed our carboys that collect the effluent from this spout on ice, and we began collecting our methane in triplicate with a gas analyzer probe. In our sampling days, we collected everything I previously mentioned, and we've started analyzing all of that. So statistically, you don't really need to know too much about this, but we used a proc mix procedure, and we compared our treatments with our periods and that interaction to see if there was anything going on. So moving into our results, this is our VFAs. So at the top you have your treatments: orchard grass, alfalfa, red clover, millet, Sudan. And we looked at our total individual and our ratio of acetate to propionate, and then acetate butyrate to propionate. So looking at our total VFAs, they do seem pretty low compared to what we would normally see, but this is still in accordance with some of our other pasture-based studies and that we've seen. And it may be that these are semi-low because of what what fluid we're getting out of the fermenter. It's not pure rumen fluid at this point. So then we looked at acetate, and our individual acetate values were greater compared to both propionate and butyrate, and it pretty much follows that 70-20-10 ratio I mentioned previously that's normally seen on pasture-based diets. And then we looked at our acetate to propionate ratio, and this ratio is somewhat high. Usually we want to see a 3-to-1 ratio, and this is showing a 4-to-1 ratio, but this still aligns with some results from other studies that were pasture-based. And this ratio is important to us because it can, as this ratio decreases, we can see a decrease in methane, and in turn, energy retention increases. Oh, goodness. Okay. Sorry for the technical glitch. So across the board, when we looked at our treatments, there was really no significant differences based on treatment. So our VFAs did not differ from each other, whether it be your total, your individual, or your ratios. So this is looking at our pH, methane, and water-soluble carb data. So for pH, we collected mean, minimum, and maximum, and then we collected methane on a milligram per deciliter basis, and then water-soluble carbohydrates on a gram per kilogram dry matter. And our pHs, if you're looking at a TMR-based diet, you think that these are kind of high, but based on pasture-based studies, these are pretty normal for pHs. So they look pretty healthy, but there were no differences between treatments at all across the board. However, our methane as a result of treatment was different. So our binary mixture of just orchard grass and alfalfa produced almost double, triple, quadruple methane as compared to the treatments, our tri-mixtures that contain red clover, millet, and Sudan. So we could potentially be seeing differences because there's more diverse metabolite profiles in these three than compared to just an orchard grass alfalfa mixture. But again, that's not the whole story. What we think may be happening here is the water-soluble carbohydrates may be impacting our methane concentrations. So based on treatment, there were differences between all four of our treatments, with our orchard grass alfalfa, which is again our binary mixture, producing the least amount of water-soluble carbs. And what this could indicate is that this binary mixture may not provide as much energy, or it's not as captured or utilized as efficiently as more diverse forage mixtures. So, because if you remember, as your water-soluble concentrations decrease, your methane concentrations will increase. So, what we can conclude based on what we know so far, because we're still waiting for information from our proteomic data, we could possibly infer that diverse forage mixtures with inclusions of warm season annuals could provide benefits that relate to rumen nutrition and energetic capture. And furthermore, the inclusion of these warm season annuals may improve energetic capture by mitigating methane production. So currently, we're still awaiting analysis for our feed and dry matter samples, and we're also awaiting everything for our protein analysis. And we did just get the proteomic data back today. It's undergoing a blast analysis right now, and then we do have PPO data, but it's also undergoing analysis. And based on this, we realized that a 2 to 3 forage mixture isn't exactly as diverse as it could be. So we're going to do a bigger study, a continuous culture study, and we have a combination of 6 or more forages that we're planning on including that are different from what we have now. And we're going to look at the same parameters that I discussed today. So I want to thank everyone that was involved with this, my committee, Dr. Sabrina Greenwood, Dr. Heather Darby, especially Sarah Ziegler for getting out there and harvesting forages with me, and all my rumen fluid collectors, and everyone at the research farms. And the funding is provided by my OREI project. And with that, I'll take any questions.
Great, thanks, Miriam. It was wonderful to have an update. And that's a great photo right there. Okay, if there's any questions for Miriam, I see there is one. Did you measure the ESC in the samples?
We are waiting for that data to come back. So we did measure it.
All right. Okay, so Kathy, I'll have you put your slides up, and if there's any other questions for Miriam, we'll see. We can always type them in the chat box, and Miriam, if you see something pop up, you can answer it, or we can, hopefully, I'm not sure if we'll have a lot of time at the end for questions, but yeah.
I'm getting there.
Yeah, no problem.
I will tell you straight up front, Heather, I'll turn my camera on if you want, but if I start to break up, shut my camera off, because my bandwidth sometimes does not appreciate.
You can leave it off unless you really,
okay.
Unless you want it on. I don't have.
I don't need it on. I know some folks prefer it, some don't. Yeah. But I know I tend to break up after it's on for a while.
Yeah. Okay. And yeah.
I'm trying to start this. Everything's working slow right now. Are you seeing anything yet?
I see your slides, but they need to go into full
Yeah,
presentation.
it's working.
was.
That's what my computer's doing right now.
Yep.
Oh, it didn't do it. Oh, there it comes. You tell me when you see it. I'll let it catch up first.
Yep. See it now.
All right.
Okay.
All right. Well, thanks everybody. We've had some great presentations, and I'm going to talk today about a project that's ongoing because it's part of our, first of all, I'm Kathy Soder with USDA-ARS Pasture Systems and Watershed Management Research Unit, big long name. We're located in University Park, PA. We're co-located on the Penn State campus, but we are a federal agency. For those of you who don't know us, we do a lot of grazing research. I'm an animal scientist there, but we try to do a lot of whole systems research. And so a lot of what we're doing lately is integrating ruminant livestock and cropping systems. We're trying to integrate livestock, pasture, and crops. And so I want to talk about one of the projects we have going on. I have some preliminary results and observations to show you what we're doing that's looking at interseeding forage crops into corn for grazing. And this is part of a large national USDA effort called the Long-Term Agroecosystems Research Network. And so there are other locations who are doing somewhat similar work, or we're doing some common experiment with grazing and with some alternative forages to try and get a better understanding of what happens across climates. So when we first look at livestock cropping systems, why integrate? Because when we look at things, it's much easier to keep things separate. It's easier to monocrop, whether you're monocropping a crop or whether you're monocropping animals versus a crop. It's just simpler to keep them apart. It's less management. So because you do need those greater management skills in order to integrate it. So if we're going to integrate, what's the incentive? Hold on, it's probably going to jump 3 slides now because I touched it. There we go. So when we're looking at livestock cropping integration, we have greater pressure to produce more food on the same or even shrinking land base because we have a growing human population. You know, you see all these numbers of how many trillion people are going to be here in 2030, 2050, and how are we going to feed them? At the same time, our farmland keeps shrinking. So we have to become more efficient. But at the same time, we have to do it in such a way that we're not hurting the environment. So we can't just keep taking more from the land without putting back. So we need to develop integrated strategies for both crops and livestock to increase the sustainability of the whole farm system while maintaining or improving the environment. And this is what has been termed as sustainable intensification. Intensifying our production, improving our production and efficiency, but doing it in a sustainable way. So for this, we need greater productivity of both enterprises, the cropping and the livestock, but we have to have that long-term sustainability. We're trying to generate additional feed for our livestock, especially low-cost feed like grazing systems, so that with the cost of feed getting higher, the cost of fuel getting higher to conserve the forages and feeds, transport them and feed them out. We also want to conserve soil and manure nutrients. We want to utilize them, not lose them into the water, not lose them into the air. We're trying to reduce nutrient losses relative to specialized and separate crop and livestock systems. If one's taking from the land, the other may be putting back, how can we put them together? And of course, the bottom line for many farmers is to improve farm profitability, because we have a lot of farms, particularly in the Northeast, that are landlocked or have other restrictions. They can't access more land. So if they need more crop, more forage, more grain, more whatever, they can't just add fields. They have to be able to do it on the same land base. So we want to increase our output, whatever that may be, if it's the forage, it's the animal, whatever our output is, while minimizing the environmental impact. So we work with grazing systems. So when we look at our typical forages, which are perennial grass-based pastures, they may have some legumes in them, we have a curve that looks like this, a growth curve. In about this time of year, late March, early April in our region, further north could be a little bit later, we'll soon start to see things green up. And we get a peak in midsummer. We have a lot of forage produced then, and it may be much greater than what our animal requirements are. If our animal requirements are only at this green line, we have this excess forage that we must do something with. Usually we harvest it and utilize it later. But when we hit summertime in our neck of the woods, forage production decreases in these cool season perennial pastures, our orchard grasses, our brome grasses, any of those types of forages, because the weather gets too hot for them and it typically gets pretty dry. But our animal needs are probably pretty constant. So we still need to make up that gap, either with another forage, additional fields, reducing animal numbers, or maybe feeding that feed we harvested earlier in the season. And then in the fall, we get another growth, hump of growth, but not as big as the spring flush. So we can maybe meet our animal needs then. But we have some gaps in this production. First one being that midsummer gap. And some of our research, which I'm not talking about today, but we are doing some research looking at some warm season annuals to fill in that gap. When these cool season perennial pastures decrease in production, can we fill in that gap with the warm season annuals? But what I really want to talk about today is either end of that scale. How can we extend the grazing season on both ends of that scale using some cool season annuals? And this is where we got looking at combining some cropland with these grazing systems. So the project I want to talk about today is looking at interseeding forage crops into corn for grazing. And, you know, this is particularly important for us when we harvest corn as grain in late October or November, because we can't establish a forage crop in central Pennsylvania. But for those in a northern climate, this may even apply for those who are harvesting as silage, because you may not be able to establish a forage crop after that harvest. So what we're using with this is developed, it's at Penn State with Penn State Extension. Jessica Williamson and I were doing this project before she moved on to another career, but we were using an interseeder. And what the interseeder does is three things. It can plant a cover crop between the rows of corn. It can apply a side dress of nitrogen for that corn, and it can apply a post-emergence herbicide. Now, of course, you don't have to do all of that, especially if you're in an organic system, but you can plant that cover crop and then apply something that if it's available to you, but this is just what the interseeder will do. So we're looking at interseeding these corn crops, and here you can see it in action. It's basically a modified no-till drill if you haven't seen it before. And it helps by interseeding these crops, we can help reduce soil erosion because we have that forage on the ground. We can enhance soil carbon. We're putting down that root mass and sequestering some carbon. We can reduce drought stress because we have a root mass to allow water infiltration, and we also have some cover on the ground to reduce evaporative issues. Suppressing weeds with that cover crop. We can hold nutrients over the winter because, as I stated, with this project, we're looking at harvesting corn as grain end of October, early November, and then grazing after that point. So we can't get another crop in to grow at that point once that corn's harvested, so it has to already be there. We can make nutrients available for a cash crop the following year if they're going to rotate into another crop. And of course, provide supplemental forage, whether it's harvested by mouths or by mechanical means. So looking at some research that was conducted at Penn State by Greg Roth, where they looked at corn yield on the y-axis and cover crop biomass on the x-axis. And they looked at no cover crops, so just a corn, red clover, interseeding annual ryegrass, or a combination. And you can see that there was a substantial boost obviously in biomass because you had a biomass from the cover crop planted, but also in corn crop, with red clover being the lowest and ryegrass being fairly similar. But when you put the two together, you got kind of a symbiotic relationship, which we see in pastures and everything else. And the red clover was probably fixing some nitrogen, but not producing a ton of biomass compared to the ryegrass, which did produce some more biomass. But when you put the two together, you got a significant bump in biomass. You're looking at 1,500 pounds per acre biomass available for harvest. And plus you got a substantial bump in your corn crop yield. So we looked at what would happen if we plant this ryegrass, which we started with, into the corn and evaluated grazing it late into the season and possibly in the springtime as well. So we're trying to extend the forage production and grazing season through the utilization of harvested or grazed winter annuals and corn residue. And this can play back into what Miriam was talking about, the pasture requirement for organic ruminant systems, where they have that 120 or 150 day minimum grazing time. And this can help extend that if you can't get it in the summertime or other times of the year, if you have a drought and you need it to extend that season, or just frankly to decrease feed costs and keeping them out of of the bunk for another month or two. So if we go back to that graph that I had earlier with all those different forages. So what we're doing here is we're planting or interseeding this forage in early June, and we're interseeding at the V4, V5 stage of the corn. So it's about knee high. We don't want to get much taller than that because we start knocking the corn over a little bit with the interseeder itself, but that's a good time to do it. We're planting our corn late April or early May, depends on the year. So if we plant it in early June, we're harvesting it as grain late October, early November. And then we're allowing the forage to grow for a little bit. And in many years it will. And it did this year, even though we had an early frost in September, it continued to grow. So then we graze it late into the year, into the fall. Sometimes we can graze it again in the spring. Now the question with grazing in the spring is whether, one, it overwintered, whether the forage took off early enough before corn planting season to get onto it, because once corn planting season hits, we need to get out and plant the corn again if we're going to put that back in, or beans or whatever we're going to put in. In this case, we're replanting corn year after year. It's a long-term study, and we want to see what happens to corn long-term if we continue this methodology, not rotating into other crops, to see what happens. So again, the ryegrass, and we used annual ryegrass to start with. We switched to cereal rye, and I'll explain a little bit why later, at the corn's V4, V5 stage. And we used at first 102-day corn at 26,000 plants per acre. This is a little bit lower than what you sometimes plant in just a cornfield, but that's important because it does allow for some more sunlight and less competition from the corn. But again, you look, the ryegrass was put in at 25 bushels per acre, but the corn yields were good, typically 120, 150 bushels per acre. And these sites are over, for those who are familiar with Central Pennsylvania, the Ag Progress Day sites, outside of State College, Pennsylvania, near Penn State. This is located there. So there's some Hagerstown soil there, pretty good soils, pretty fertile soils. But this is what we were seeing on average. So in 2017 was the first year that we grazed. And we did use annual ryegrass that year. We had 60 dairy heifers come in. We had grazed and non-grazed plots for a control. We had 2 sites about a mile or so apart, and we had 3 blocks within each site. So we had 6 different groups of heifers, 10 heifers for 6 groups. They came in on December 11th and left on the 21st of December. We did have weather issues that year. When we were getting into December, we did have some very, very cold weather, which affected our ability to keep open water to the animals. Because if you look on the bottom right-hand side of the screen, that black dot is a Rubbermaid tank that we would bring water in on the back of a truck. So we did not have electricity, we did not have running water. So we had to rely on weather to keep the tanks open. And when we got some cold weather during there, we were out breaking those tanks open several times a day. We did use the 102-day corn. And what we learned that year is that we grazed too late in the fall, because it was too late. Because the other thing we had over and above the temperature is we got some very, very heavy snow cover. We had a snowstorm. And while animals can graze through snow, when it snowed and then got cold, we got a really hard crust on the snow. And that made it really difficult for those animals to get through. Additionally, these animals, dairy heifers, were not used to grazing in these conditions, so weren't real happy about it. We did end up putting a little bit of hay out for them a couple of days because we were afraid they weren't going to be able to eat enough. It's imperative to have access to some type of shelter or wind cover. It's going to depend on the animals, but these dairy heifers really did need some type of shelter or wind cover while they were out there. And the hardier the animal, obviously, the better the system will work late into the year. So with these animals that we used, they were not from a grazing farm. They did graze as heifers and dry cows, but they weren't from a grazing farm, so may not have been the best adapted animals, but were the animals we had available at the time. And so we thought maybe a couple of things we could do is graze earlier and also try a shorter day corn so that we could get on a little bit earlier as well. That year, we didn't have much regrowth of the ryegrass in the spring until it was time to plant the corn. So we had this heavy winter and then a late spring, so we did not get the graze at that second time. So what we changed in the second year in 2018, we changed to cereal rye instead of the ryegrass. It comes on quicker, earlier in the springtime. It had a little bit longer growing season than the annual ryegrass, and I think we just thought maybe it would make a better fit. We used a slightly earlier maturing corn. and grazing a little bit earlier in the year. We've been grazing somewhere around mid-November, letting the grass grow about 2, 3, 4, sometimes 6 weeks, depending on when the corn gets taken off, instead of mid-December. And we graze beef cows instead of dairy heifers. We found a local herd who was willing to bring their cows in and graze them. And these cows were used to grazing standing corn and corn stovers. So these girls came in, dropped their heads, and went to town. They thought they were in heaven when they saw that green grass. So it made a huge difference in not only the utilization of the forage, but also the condition of the animals, because these girls actually looked like they gained weight. We couldn't weigh them, but the owner thought that they actually put on condition while they were out there, whereas we know the dairy heifers were losing a little bit of condition. So in that 2018 grazing season, we grazed in the fall, and then we were able to get back on in the spring. And you can see the growth that we had in the springtime. We brought the same number of animals on in the springtime because that's what he had available, but we probably could have used 50% or maybe double the animals that we had because we couldn't keep ahead of it. Alternatively, you could harvest it as well, but we wanted to put it into a grazing system. So we had the 24 mature beef cows again, and again, but we just didn't have enough grazing pressure on it. 2019, I don't have data on. There were some personnel changes and we're still going through that, but I wanted to show you some pictures from this past grazing season in November 2020. We had, again, 24 mature beef cows. We grazed November 16th through December 4th. Now this was a drought year. For those of you in this region know that we were very, very dry this past year, but we had some really good growth in that ryegrass, or that cereal rye, even despite that. Now our corn yields were affected. The one site had decent corn yields at 120, 125 bushels, but the other one just a mile down the road, which probably isn't probably as well watered or have as good of a soil, was low in corn yield this year. It did affect that. But when we looked at our total available forage for, which would be the rye plus the corn stover, these two sites were very similar. And so they had some good available forage for these animals to graze into the late fall. So if the back up, I wanted to back up there. I wanted to show you that top picture there. When we were pulling the cows off, because we did strip graze them, and we did not back fence them because they had to come back to the same water source. And forage really wasn't grazing actively like it is in the growing season. So we didn't feel a need to back fence them. But the day that we took the cows off, that top picture is from the very first paddock they would have been in about 10, 14 days before that. Even in late November, we took them off after Thanksgiving. Late November, you can see there was a little bit of regrowth on that rye, even at that point. Now those cows weren't coming back and actively regrazing that like you see in the summertime or the springtime whenever they get into the green lush forage. We didn't see them come back for a drink and right back out. So they hadn't run out of forage to the point where they were regrazing that little bit of regrowth. So if we take a look at the 2018 data, which is the most recent full data set that we had about 4,950 pounds, just shy of 5,000 pounds of dry matter per acre if there was 100% utilization. This included corn residue and the ryegrass, or sorry, that would have been cereal rye that year, 2018. But of course, we're not going to have 100% utilization. So if we get more realistic with a 50 or 65% utilization of the forage, you're going to have about 25 to 3,300 pounds of dry matter per acre available to those animals. Just the ryegrass alone, without the corn stover, there were 1,750 pounds of dry matter per acre at 100% utilization. And you can see where it is at 50 and 65% utilization. So still, 1,000, 1,100 pounds of dry matter per acre available to those animals. So what does that mean to a beef producer? If we pick the 65% utilization, if we have animals consuming 2.5% of their body weight, and we have 30 cows, beef cows at 1,200 pounds each, we could graze that herd an additional 73 days on 20 acres with a 65% utilization. With the 50% utilization, it's similar. We had 55 days, a little bit less because we're going to leave a little bit more residue out there on the 20 acres. So at what cost savings is that? If you take $2, $3, $4 per day, if you're feeding hay, feeding silage, feeding something else, what's the cost savings to a farmer if you can graze another 2 months? It'd be significant. And then what's the value of that spring grazing? We haven't been able to do that enough yet, enough time to know what value there is. And we're still learning how to handle that forage because there's so much more forage in the springtime. So that's our ongoing project. We are actually starting to watch that forage now because we've had some fairly warm weather to see when it starts growing so we can get on it, to try and get on it before we have to plant our corn in late April, early May. And stay tuned because we want to continue this project and actually evolve it a little bit because we're just using the ryegrass, or I'm sorry, we've used the cereal rye right now. We see advantages to using and adding things like legumes, maybe some red clovers, maybe some brassicas, some turnips or something. But we wanted to start out simple so we weren't confounding our results, but I can see us adding some of those components later on. So I'll stop there.
Great. Thanks, Kathy. That was awesome. Yeah, we're doing much similar work as we've chatted about and hoping we get more chances to work together now that we're certainly having a USDA-ARS center here in Vermont too. We do have a question here. Was there significant reduction in corn yield in 2019, 2020? Sorry, they missed it.
Yeah, the 2019 I don't have data for because that's when Jessica left and her data got transferred to us and it's still sitting on a computer. 2020 it was. In fact, you can still see my screen, right?
Yep.
Okay, right there was our corn yield from 2020. We had a drought year. And so yes, the one site was significantly hit in corn yields, about half of what it normally is, 65, 70 bushels. The other site was still pretty good. I think there might be some more springs and water activity pretty close to the surface on that second site.
Great.
Our rye and corn stover yields were very similar. You know, the rye was very similar in yield. So it was a difference in the corn, but the rye was doing all right.
Super. All right. Well, we are at 2:02. We're right on time and appreciate everyone attending today. And it looks like most of the questions were answered in the chat by the various speakers, and John Barlow did just step off. So if anybody has questions for John, he says, please just feel free to email me, and I'm sure our other speakers, Andre, Kathy, and Miriam also, I'm sure, are open to receiving any additional questions. Thank you. Thanks to our speakers today. Really appreciate your time. It's so wonderful to hear all the great research that's happening all around us. Never feels like we have enough time to to get it all out there, but you know, you're always welcome to email folks and stay in touch. So just want to invite everyone to our last dairy webinar tomorrow that will be focused on corn silage hybrid testing and management tips for the Northeast, being presented by Joe Lawrence from Cornell Cooperative Extension and the Cornell Pro Dairy team. So with that, I will say have a great day. And again, thank you to our speakers. Appreciate your time, and thanks for everyone for taking a little time out of your day to spend with us all. So with that,
Thanks everybody.
Thank you.
Thank you.